Yeah, so good afternoon, everyone. Can you hear me?
Yes, we can hear you clearly.
Oh, thank you. So yesterday we introduced another topic titled biological databases or databanks. So we said quite a lot yesterday, and we also got to showcase, I mean, live what NCBI looks like. So we said a lot of things. We said NCBI is a composite database. Then we explore the NCBI, not fully, but just to give you a taster of what today's class is going to be about. So the plan yesterday was just to to lay the foundation for today's class. So we're just trying to talk about theory and some terminologies. And then I sent a document to the WhatsApp group, which I'm hoping you guys have had the time to go through it. Please, you have to go through it because there are a lot of things that I didn't say in the class, but are very, very relevant to you. So, and if you actually read that document, it's not a long document, just 10 pages. So we can just dedicate probably 30 minutes. I think we should be done because it contains literally what I said in the class. So, I mean, if you watch the video very well and you grasp what was taught yesterday, so I mean, understanding the documents, the PDF documents should not be, I mean, that's difficult. So I'm talking about the second PDF document I sent to the group, not the one, I mean, the slides. So today we are going to demo NCBI. So we're going to have some practical session on NCBI. And our objectives today are this. So we'll be showcasing how you can use NCBI to do basic literature search, and we'll also be showcasing how you can, I mean, use NCBI to search for probably, I mean, gene sequences or any information regarding the gene or protein. So, I mean, those are the two things that we'll be doing now. I must also say that, I mean, there are lots of things that you can do on NCBI. There are a lot of things, and then we won't be able to explore, I mean, the full potential of NCBI in this class. So, but I will just focus on these two for now. If you actually want to delve deeper, so there are tutorials that I'm going to point you to. And then, so I think there will be a lot of assignments so that you guys can actually get your hand dirty regarding NCBI. So for some, I don't think this is the first time they are experiencing, I mean, they are interacting with NCBI for some people. Why for others, I'm thinking probably this might be your first time interacting with NCBI. So let's look at what NCBI looks like. All you just have to type is just type NCBI and then click on this. So this is the NCBI homepage. So what I did yesterday was just show you, I mean, a lot of things. So, I mean, so if you want to have access to everything, I mean, all the resources that are present on NCBI, I mean, just click on all resources and then you see a whole lot of things. So, I mean, a whole lot of things. Some are tools, some are, I mean, databases, some are, I mean, so these are the tools and these are the, I think, we have quite well, these are the databases. So everything has been categorized for you. And if you need to submit your genome as well, I mean, you just probably click on this pane. Anything you want to submit, probably metadata, any sequences, if it is 3D image structure that you need to submit. All you just have to do is come to this section and then click. And if it is tools that you are interested in, I mean, these are the available tools that we have on NCBI. So today we are going to be talking about how you can retrieve. You know, I mean, when I was telling about NCBI, I said NCBI you can deposit, you can store data there, and you can also retrieve information or data from NCBI, and then I mean use it for your own personal use. So you can fetch, you can download, and you can do a whole lot of analysis. You can upload as well. So today we will just be showing you a whole lot of things. So if you click on all databases, so you can see these are the available databases that we have. So it's either you find the available databases here. So you can either find it here or you can come here again to all resources and then click on databases. I mean, those are the two approaches that you can probably use to have access to any database. In fact, majority everything that you find here, you can also find it here. So it just depends on your own, taste. So what you want to do. Then you can also probably find them here. Probably, if it is DNA that you want to work with, just click on this, and then you have the whole access of everything. Everything has been categorized for you so that you don't have to, stress yourself locating things. So everything has been well organized for you to be able to interact with. And if you need tutorials, so you can also come here. So this is where you have your tutorials. NCBI toolkit manual, NCBI anything. So tutorials on anything, I think you'll be able to like find it. So I'll go back to my role. So today we are going to be exploring the on-trace search engine. So on-trace is spell this, and this is how you pronounce it, on-trace. So this search engine that you are saying, it is a very powerful engine, and this is what to allow you to retrieve any data or any information from Instagram and this particular search engine. So just see it as Google search engine. If you want to search anything, you have to come here and then search for anything. So you can search for anything here. But the analogous version of that on NCBI is the on-trace search engine. So it is different from Google, although they have, I think they have the same framework. They have similar framework, but this has been developed. This has been optimized to work very well for NCBI. Yeah. So, and like I said, we have two objectives. And number one objective is we'll be showcasing how you can use NCBI for literature search. And this is because as a researcher, you will need to like read a lot sometimes. You want to publish, so you want to stay. You want to be current about the latest development in your field. You want to be able to like retrieve some information, or you probably want to perform an experiment. You need to like learn some methodology. So you will need to come to NCBI. And then from the NCBI, you can see we have a lot of stuff. So we have, these are the databases that we have. So these are the databases that we have here. And the information that we'll be searching. So it's going to be probably through the PubMed. So PubMed is a created databases that contains information about articles. So anything that has to do with articles, publications, anything that has to do with articles and publications. So you'll find it on PubMed. So, and if you actually, let's say probably want to search for, let's say anything like probably black oxide gene. Okay, don't let me use black oxide. Let's say probably stress or heart attack. Heart attack. So all you just have to do is type this and then search. So by the time you do that, you can see. So it was able to find the following information regarding heart attack on this particular database. On Bookshelf, it was able to find like 20, like at least 20,000 instances of heart attack in Bookshelf database. But what we are interested in is actually the PubMed. So the PubMed. So if you click on the PubMed, so we can see. So these are all the instances of heart attack. These are the articles that probably has anything that they talked about attack in the article, whether in the title or in the body of the article itself. The introduction or the discussion or the methodology. So, these are all of the articles. So, you can then probably click, let's say probably you are interested in anything, and you can narrow down as well. Look at the slider, you can narrow down if you want. So, let's probably, so it's like the first instances of heart attack probably started around 1814. But you can see a lot of interest. We have a lot of research on this particular heart attack. Starting around probably 1990s, 1980, you can see this So I'm not sure whether I can see this pic. But let's probably are just interested in research, recent uh, publications. So you can just probably use this slider and then probably fit out. Probably are just interested in recent. It's probably between 2020. Yeah. So you can just use this slider just to limit your options. So, and you can see the everything that's reduced from up to like probably 3 million, now have like 154. So these are very recent. You can see 2025. So, and if you like, you can also probably click on this to select and to narrow down. And let's say probably you are just interested in probably you are not interested in the abstract, probably you are just interested in full text. And check this box and check, right? And then probably what you're interested in is a review or clinical trial or books or anything, you can probably do that as well. So let's just click on probably review. You can see we now have 21 instances. That meets this particular, filtering. So this is actually how probably you search about anything. But this is just very basic. So the example we are going to be doing is we are going to be exploring the use of booleans because this is just art attack, but probably you are just interested in some other things as well. There's probably art attack in adults. In adults, probably if you do that, you can probably search as well art attack in adults. So it's going to do this. And for the best way to actually do this is to actually probably combine it with booleans. And the booleans we have the and or anything that's okay. So we have this and for booleans we have and, we have or, and we have not. So using these Boolean operators, we can actually combine a lot of things together. So, and one way to actually do that is you can probably art attack and adults and you can search. So you get 5,820 records that meet these criteria of art attack and adults. And probably you are interested in recent stuff, you can click and use these sliders to narrow down. And if you need to like add more features, you can click here. And let's probably are interested just in English articles, click on it. So, and then any other thing that you want to add, you can add it here. So, and anything. So then it depends on you as a researcher. So what you actually want to do. So this is just like a taster of what we are going to like do today. Now I'm going to take a much more formal approach to actually using the entries search engine. So like I said, if you want to get to this PubMed, there are so many ways that you can get to it. I'm trying to like go back. So if you click on this label, so you should be able to like go back to the NCBI homepage. So instead of probably searching broadly, you can probably just click on PubMed directly and then search for art attack here. So if it's do art attack and then search. So you're just going to like narrow the search alone to PubMed. You know, initially what I search, you can see this. So it just took me to PubMed directly. The other way to if you remember what I did previously was this. I'm going back to NCBI. NCBI. Sorry, internet. So what I just did initially was art attack, and I click on art attack. I type like art attack, and then I selected all databases, right? And when I did that, look at what happened. So it searched all the databases, but in these instances, I already limited myself that okay, just look, don't search across all the entire databases. I only want to search. I want you to search just the PubMed database. And when you do that, and then, okay, I didn't type. So PubMed, then art attack. So just compare the two. So this one took me directly to PubMed, and this one, just search through the entire databases and then since you are interested in PubMed, you can just probably click on PubMed like this. And then you can see you have 320,000, results returned. And then the same thing, see the same result but different approach. So that's number one ways to actually probably get to PubMed. So the third way to get to PubMed is just to like probably type PubMed directly. So instead of probably going via the NCBI, you can just probably type PubMed directly, and then it's going to bring you to this homepage. So this is actually the PubMed homepage, and then you can now say Art Attack. We still see that we get the same thing. So if you do that, so what do we have? Still the same thing, 320 and 2. So those are the three ways that you can use to get so PubMed. Number one is either probably first, open the NCBI homepage itself, then search, and then click on PubMed, and then or you click on PubMed directly, and then you get to this homepage, and then you do any search that you are looking for. Yeah. So, but the example we are going to be looking at today is still kind of like related to Heart Attack. So we are going to like consider a lot of ways that you can use to search. So I will go back to my slide. Yeah, I was saying like we have two. So yeah, so there are two ways that you can use to do a literature search. Number one is either you use your keyword or use the mesh term. And I will explain what mesh term means later. But let's say probably we have this task. Let's say this is the task that we have. Let's probably what wants to do is, what he wants to know, let's probably have this question that wants to answer. What is the role? I think probably I should, what role does pain have in sleep disorder? Yeah. So let's say this is our task. What role does pain have in sleep disorder? And wants to like probably search the literature for any articles related to this. This is your question. So you are the one, you frame this question yourself. What role does pain have in sleep disorder? So this is the question you want to answer. So if you actually want to do this, I know some people will just copy this. Let's even try and copy this. So let's copy this and then paste it here. What role? Okay, let me just click on this. Okay. And what role does pain have in sleep disorder? So let's click. You can see we have these results returned to us. Potential role of obstructive sleep apnea on pain sensitization on jaw function, temporomandibular. So that is actually one way that you can use to search this thing. So the second way is just break down. So break down. So all of this, this is your question. So break them down into key terms. So what are the key terms here? Can somebody tell me? Toby, please, I'm not sure whether you can call somebody. So based on this thing, so what do you think are our key terminologies? So let's write down our key terminologies.
Taiwo, can you unmute and tell us what you can tell? Taiwo, please, you are holding everybody at ransom. Abdul Basit Taiwo. Okay, I call on Oyelami Samuel.
Okay, I think we have pain and we have sleep disorder.
Pain. Exactly. So we have pain and we have sleep disorder. So we've broken down our question into what? Two key terms, pain and sleep disorder. So what will happen is now you have to copy this pain. So I'm going to like use another search engine. So do this pain. Then you want to use a Boolean pain and what? The second key term So which is this, and then you click on this. Then you click this. So let's compare our results. In the first instance, we have 1,116, and this one we have what? 10,112. So this was able to like capture more things because we were specific. You know, I mean, this one is too specific. If you actually think about it, what role does pain have in sleep disorder? So if you're just trying to like probably, so it was not really, I mean, it's I mean, it's not really that. How should I put it? I'm trying to like look for the right word. So it's not really doing what we want. So you can see that we have fewer hits returned compared to this. So it's possible that we've actually probably left out some valuable, I mean, articles regarding pain and sleep disorder by the time we do this. So whenever you are trying to search, unless probably you know the topic, I mean the specific article that you want to read. You know, there are some instances where probably you will have like an article that you want to read, you have in mind. So you can just probably copy everything verbatim and then put it here. So it's just going to like restrict you to that particular article. So, but let's say probably want to like just want to like probably explore. I mean, want to read about, I mean, pain and sleep disorder. So, I mean, probably doing this is not the right way. So, doing this kind of approach is not the right way. So, you want to be using, I mean, you want to break down your question into key terms and then combine them with operators. And if it's probably it is all cases that apply for you, you might probably put it here. Let's probably are just interested in articles that talk about pain or sleep disorder. So you can click on this. You can see you have more, I mean, more hits because you have like a combination of, you have articles that contain either pain or either sleep disorder or combination of both. So that's why you have more, I mean, it's on for you. So, but in our own case, what we're interested in is articles that talks about both pain and sleep disorder. So, and then, so this is what we have. So we've been able to like capture more. So psychosomatic aspect of neurology and things like that. So that is actually one way. So this is, okay. This is how you do, I mean, you approach this question. I mean, with some, I've shown you two ways that you can actually probably use to do this search. So the other way is to actually probably take use of the advanced functionality. So if I should click on this again and then click on this advanced. So this is where the on-trace search engine actually becomes, I mean, very powerful. So you can then start nesting everything together. So the same thing that you've done, you can just probably put, okay, let's say probably, so you can see, I'm not sure whether you can see this. Look at my cursor where we have this all feed. So this was, okay, let me just show you this one first. So what I typed previous, you can see them here, pain and sleep disorder and everything, you can see them here. And you can see how the search engine interpreted everything. So when you put sleep and disorder, what was actually wrong is this. You can see it was actually doing some interpretation behind the scene that you guys are not seeing. So when you type everything, it's interpreted it as this. And look at this one. What role does pain disorder do? So let's see how it was able to interpret it. You can see. So you can see row, mesh terms, or row. So this was what was, it broke down all of these things into a language that it understands. And then it's now analyzed on the needs. So, but let's see how we can actually probably make use of the advanced functionality of the on-tray. So yeah, let's say probably it's still the same question wants to answer the role of pain in sleep disorder. Remember, but let's say you now want to limit, don't want to search across all the field because by the time you search, I mean, when we did this thing, when we did pain and okay, this is how we combine it, pain and uh, and sleep disorder, sleep disorder. So you know this was what we typed. So when you type this, so it was, so when you run this, it's going to search across all the feed if you run it. So it's going to search across all the feed. but we don't want to search across all the fields. Let's probably, you just want to search because you can still look at all the fields that we have. So it's just okay for the instances of pain and sleep disorder in whether probably it occurs in auto or auto corporate or like date or anything. So these are all of this. It was searching across all of this field. So when you click on, if you don't limit your option, but you can actually probably limit your option that, okay, I don't want it to search across the entire field. So I want it to thus look for the instances of pain in the title. Let's probably I'm just interested in the title. So let's say probably title. So let's probably just want the search engine to return only the instance of sleep disorder and pain if they only occur in the title. So what we just do is this. So you click on sleep. This okay. Let me first put pain. Put add and title and the sleep disorder rather sleep disorder. So when you do this, put add. So what is just going to happen? You can see the interpretation has changed. You can see before it was this pain and this, but now we don't want to search across all the entire field. We just want to limit our attention to what to a specific field. So this one is only going to like look for all instances of pain and sleep disorder if they only occur in what's in the title. And when you run this, so let's see what will happen. So we can see we now have less results, and we can see we have pain and sleep disorder in the title, just in the title alone. So they only occur in the title. So that's actually what is happening here. So we can see we have less searches. Yeah. Please, do you understand up to this point? Before I continue, do you understand?
Familiar, do you understand?
Yes, I do.
Others,
Clement,
do you understand up to this point?
do you understand? All right. I think we can continue.
Yeah, I think I lost my line of text. Okay. So let's talk about this before. Yes. Okay, let's say probably now you can let's probably you like a particular author or you want to know some articles that have been published by a particular author. It's also possible to actually probably make you, exploit the on-trace search engine just to make you acquainted. So how you can actually look at it. Just click on the PubMed and click on advanced. So you come to this page. Let's probably I want to see all the articles that have been published by Prof. Iruka Okeke. How can I do that? So what I will just do is click on author and then click on what Iruka Okeke and add. Then you can search. You can see these are all the you can see Okeke Iruka. So this one I think this is our latest publication. So factors associated with positive blood in children. Yeah. So let's say probably we can also find my name. So I doubt it. I don't have any publication. So Habib. See my life.
See you alive.
I have a publication. So you guys are welcome to read it. So you can see my name, Habib A. Adeni. So yeah. And you can see Tochukwu Adbuisi as well. So and you can see Oluwato Biloba Kasi. So you can say Iruka and Okeke and others. So yeah, if there's a particular, let's probably, you are following a particular scientist and you want to be, you like what he does. So you can actually filter, just limit your search to a particular field. So and these are the list of fields that you can see. These are the list of fields. Just explore if it is a particular journal, if you know the volume of the journal, just probably use this. And if it is mesh term that you want to use, if it is the investigator, anything that you want, so you can filter and narrow your. So it's just similar to you using your Excel, So let's there's a way you can filter in Excel. So if you let me show you one Excel that we can work with. Yeah. So it just says, okay, so these are the few, like all of these things that you are seeing here, all of these things that you are seeing here, all of this stuff, just they are analogous to this. They are similar to this. They are similar to the columns, ID columns, resource column, back inspector columns, back inspector, this. So they are same, just see them as columns. And then, you can now filter based on this, value of a specific, of a particular column you can filter. So, and one way you can do that is, okay, let me create filter. Let's probably want to see, this is everything. We have, how many records do we have here? We have 103 records, right? Rows, 103 rows. Let's say I want to filter it based on, I'm just interested in everything that failed. I can just do this, and you can see I now have like how many entries I have? 60, I have 17 called after filtering. So that is actually what is happening here when we do all of these things, when you select. So you just want to filter based on specific columns, so specific fields. So in this instances, we are using field. So, but just say field as analogous to your your column names in your Excel sheet. And just like you can filter based on a specific column, you can also filter your search, so that you are specific. You actually want to be specific, so you can just narrow your search to specific columns. So that is another way to actually search for things using the on-tray. The last way, so the other way is going to be the mesh. So the mesh term. So mesh actually means medical subject heading. Medical subject heading. So, and let's see how you can actually get to mesh. So, let's just talk about mesh first. So before that, let me just click on mesh database here. So, or we can even come here and read about mesh. Let's see whether we can get information about mesh. All resources. Let's see mesh, so that we can see. Okay, so it's saying mesh. So let's read about mesh. So from the mesh database, from NCBI description. So mesh, which means medical subject heading, is the U.S. National Library of Medicine controlled vocabulary for indexing articles for Medline or PubMed. Mesh terminologies provide a consistent way to retrieve information that may use different terminology for the same concept. So the idea behind mesh is this. Do we have any Yibo guy here and any Hausa? So in Yoruba, okay, let's say probably we are talking about cats. Is it cats? So this is the English name for cats. So this is the English for cats. In Yoruba, we call it Olondo. I can spell Olondo. Tochuk, what do you call it in Yibo? Tochi. But the idea behind it is
Ask Kelechi is here. Victor is here. Let them answer.
and Victor that does not. Is it Victor in our lab or Victor?
Yes, Victor in our lab.
That one does not understand Igbo.
Hey, Jesus. Kelechi, please move in.
I think I can. Let me go.
I don't know too.
Hey, wonder.
It's millionaire here. I think millionaire should be able to.
All of you are not Igbo. Don't even claim it.
Clement is Yoruba.
No, I didn't say Clement. It's Kelechi I'm talking.
Okay. Victor. Probably Victor might know.
Even if I know it, I will not even answer it because you have already shamed me in public. So no problem.
Oh yeah, no thanks.
So in Igbo, I googled it. It is
Okay.
Wamba.
Wamba.
One
Yeah.
And then in Hausa, there is Busu. Buusu in Hausa.
So Igbo it is Wamba. I can't
Yes.
spell it.
Wamba.
Yeah. So you can see we're all talking about the same thing. So
Oh,
let
no.
me look for
That's
cat.
that is Mage, M-A-G-E.
Okay, for Hausa.
NWA.
So this is a cat. So in English, so this is what they will call it. They will call it cat. Yoruba will call it Olondo, and then Igbo will call it Bamba, right? But we are all talking, we are all describing the same thing, right? So it's possible that probably, like, let's say probably now want to deposit your, let's say probably you publish an article, and then you are describing probably sleep disorder in cats, sleep disorder. English we call it sleep disorder in cats. Probably Yoruba might call it sleep disorder in Olondo. Right? But it's still the same thing we are talking about because cats is Olondo. And Igbo will call it probably sleep disorder in Wamba. In Wamba. Yeah. But it's still the same thing that we are talking about. So that means whenever you are searching, let's probably now want to search. These are all the same thing, the same article, all talking about the same thing. So probably this one was published by Clement, and this was published by me, Habib, and we both submitted. And this one was published by Victor, Emeka. So you see the same card that we are talking about. But now if I want to search, let's say Clement wants to search, probably wants to learn anything because probably Clement is an English speaker. And then he types this in on trace search engine, put cats. By the time he puts sleep disorder and cats, all of these things, all of this, the only article that will be returned is this. All of these things will not be returned. The remaining two will not be returned. And that's because, the keyword is, Clement is using the sleep disorder and also what? And cats. The same way, let's probably also want to learn about sleep disorder and Olondo. If I should put that in on trace search engine. So what will happen is that, the only thing that will be returned is probably anything that has sleep disorder and Olondo. So I will miss out on article published by Clement and article published by Victor. Things like that. So I'm not sure whether you get the logic behind this. So what's now leads to the creation of meshes? Mesh here is talking about controlled vocabulary. So instead of probably, using different terminologies to describe the same things, so can we just have like a unified, like a controlled vocabulary that will unify everything? Such that if I want to retrieve any information in cats, so if Clement is now using sleep and cats, it will automatically get information regarding the others. So that's actually what leads to the creation of mesh. So before, if you don't use mesh, you will miss out on article published by me, Habib, and also by Victor. If Clement is the one doing the searching using sleep and cats. But if you are now using control vocabulary, let's probably now have a control vocabulary. Let's say probably, okay, we now meet okay, and say, okay, probably we should call this thing. What is the botanical name of? I think Canis felix. What is the botanical? I say botanical name. The scientific name for cats. Scientific name.
Shame on all of you that did Zoo 111. Shame on all of you. Shame.
Felis catus. I was close. Felis catus.
Shame on all of you. Shame.
So Felis catus. Where's my slide? So Felis catus. So you can just kill. I mean, let's just have a control vocabulary. So instead of everybody using their own different terminologies, okay. So let's just use Felis catus as a what? As a representative of all of these things. So what's mesh? So this is going to be the mesh term now, mesh term. And all of these entries. So it's just like, I'm not sure whether you guys are acquainted with dictionary, I mean, in probably Python or R or things like that. So it's just like probably you having this key and items relationship. So key and item relationship, key and item relationship. So your key is going to be Felis catus and your items are going to be all of these different terminologies. That's loan group. And the number. Yes. So this is going to be your mesh term, and these are going to be the entries. So just say it as okay, your Felis catus is the key and the items. So if you now want to like probably search, the better was to use. So instead of probably using cards, Olondo, or Wamba, by the time you just use probably Felis catus, automatically you will have access to everything. Whether you use the name cards or use the name Olondo or use the name, I mean, Wamba. So that's actually the idea behind mesh term. Do we understand the idea before I start demoing the mesh? You can actually search using mesh. You actually see that mesh is actually very powerful. So it is still the most, I mean, the best way of searching. Do we understand up to this point? And do you have any question before we move on? Adiola, you are too quiet. You've been quiet. You've been so quiet recently. Are you with us?
Yes, I'm with you.
Do you understand what is going on?
Yes, sir.
Thank you. So this is, so now let's look at mesh in proper. So all we just have to do is to come here again. So you just click on this logo. So if you want to go back,
Question.
please, can you go ahead with your question?
She wrote it in the chat already. You can.
Does the mesh term have to be a scientific name? No. So I just use that one as an example. You will actually see, I mean, when we demo now that it doesn't have to be a scientific name, but just like a control vocabulary. So it doesn't. So yeah, and go on. So instead of probably typing, if you want to go back to NCBI homepage, click on the logo. And if it is PubMed that you want to go back to, you can click on this logo as well. So you don't need to be probably be typing NCBI in PubMed on your Google search engine. So remember, there are two ways that you can actually, let's probably now want to, we've broken down our word into pain and sleep disorder. But it is possible that probably we have other synonyms of pain. Can somebody probably tell me other synonyms of pain that you know? I don't know, but if they say probably is in pain, and you tell me probably other synonyms that you think you know. That's why I don't want to use pain. What other synonyms can I use?
Agony.
Victor. Okay. Agony. Yeah. I'm talking about, yeah, you can use agony. What again can you use?
I'm let me say hot.
No. Can't be hot. Can anybody think about any medical terms for pain? There's a medical term for it. I can't remember. But so it's possible that probably the author, I mean, based on the pseudonyms, so we can have different ways.
Tenderness.
Eh?
Tenderness. Some people say it's tender.
Tenderness. I don't think that is correct, but I shall go with you. Tenderness. Yeah. Does anybody have like a medical term? We are talking about, I mean, we are a medical student. I mean, we are a biology student. If you want to speak in biological term, you want to express pain in biological term. I've forgotten it, but I know there is a medical term for pain. I can't remember. Medical term for pain. Medical. I'm all the stuff for pain. Nociception. Okay, nociception. So let's put that.
Search tenderness as you walk to. You see, it's also a term for pain.
Yeah, okay, tenderness. So we can see all sort of pain. So let's just put C, which is association nervous system. So this one is even I'm looking for a broader term. But we shall get the gist. So for sleep disorder, can somebody tell me other synonyms that you can use for, I mean, to express sleep disorder?
Apnea. What is it? Sleep apnea.
Insomnia.
Yeah, I think insomnia. But that was the first thing that came to me.
Yeah, insomnia.
Apnea. So, I mean, these are the ways that you can actually probably use.
APNEA. APNEA. Apnea.
Okay, apnea.
Yes.
So these are the ways that you can.
EE, not IE.
So these are the ways that you can probably use. I mean, the different ways that you can use to express pain. So that you use agony, you use tenderness, you use this. But let's now see. But what wants to use our own when we broke down our own? I mean, this. Remember, this was the question we were trying to like answer, right? And we broke them down into pain and broke them down into sleep disorder. So let's now use. Let's see the control vocabulary for pain. I mean, if I actually want to like search it properly so that we are able to like get around, I mean, all of these different synonyms. So let's say the mesh term. So what you just have to do is, so there's a mesh database below. If you come to this, please, or you can also probably go on NCBI and then start from there. So that you can come here, mesh, can come in as well and then put pain and search. So you can see, and or you come here, or you come, sorry, you come here, mesh database, and then type this. So any one that you want, and then you can probably scroll around and see the ones that is specific for you. But I'll just click on this and see. So I think you should just go with. So you can read, you can see different ones. So the first one is pain, an unpleasant sensation induced by noxious stimuli, which are detected by nerve ending or nociceptive neurons. So intensive discomfort, distressful agony, and things like that. So I mean, just read through because it was able to like return 66 results. So I mean, just read and then click the one that applies to you. If it's probably musculoskeletal pain that you're interested in, click on it. If it is visceral pain that you are interested in, click on this. But we are interested in just the general pain. So I'll just click on this pain, and then you can see that the mesh term is pain itself. So you can see it is pain itself. So and under this pain, you can see we have abdominal pain. So all sort of pain we find it here. So if we use this particular one, automatically anything that is talking about abdominal pain and everything is going to be also is going to be, by the time you search, it's going to be returned as well. So, and you can see the entering terms. You can see So all of those things, all of those things that we are talking about. So these are the words that captures them. You can see suffering, physical, suffering, suffering, physical, aches, is pain, burn, agony, crushing, pain. So things like that. But we're just going to, that means we are on the right track. So that means pain is what we are going to use. So let's go back to our PubMed. Okay, so I don't think I want to even do that. So what you can just do is, so by the time you do that, you can say probably search, adds to search. So what you can even see there is you are seeing, I think this one is not supposed to be there. This one is heart pain. So myocardia. So you can see pain. You can add to search builder and then search for the other one, which is sleep disorder. Sleep disorder. Search for the other terminology, which is sleep disorder. Sleep disorder. So and just click the one that applies to you. I'm just by reading this. So which one? Abnormal sleep wake schedule pattern associated with circadian rhythm, which affect length, timing, or rigidity. Anyone that probably this is what you want. You can just probably click on this. Click on this. So I'm still going to do this because so the reason why you are seeing this myocardia is because I searched for this one previously. So and once you do this, you can add also to this. And so this is what you now want to copy. You want to copy this. Instead that you copy this and put it here and put it in PubMed like this, then search, and you have all of these results given also you. Or when you click on this, I mean all this search and adds to search builder, you can directly just click on this search PubMed and you will still get the same thing. You can see both 29 results, 29 results. So actually it just depends on you. So this is what we can see, pain, mesh, and all of these things. So I mean you can now start reading your article. That's why we now want to send, you want to send this, you can send this to yourself. I mean you can send this to yourself. But I should click on this. You should be able to like find that information. So I just need to like probably go to my mail. Then I'll see that. And see. I have all of those results. Can you see? Then I can now start reading these articles one by one. So you can send it to yourself. And you can also send to your words, to your clipboard. Your clipboard. And let's probably want to send it to somebody. You can just click send to your clipboard and then send it. Or to your bibliography. So when you do this, so you can always come back to this. So you click on this and then you click on your dashboard. So you can see that. So because I've created, so if you don't have an account on NCBI, you won't be able to like send this into your bibliography and then you won't be able to like access your dashboard. So it's only when you've created an account on NCBI. So that's where you can see all of these things. So I think I have internet. Sure. You can see. Content and items, you can see everything. So all of those things. So you can save it with, I mean, all of those things that I did. So they are here. So they are here. And then you can see, let's say you want to know about your activities as well. And you can find that information on your dashboard. And what again can we do? Again, can we do display option? So probably want abstract. Let's see the abstract format similar to me. So if you want it to be displayed to you as abstracts, you can also, I mean, just play around with these things and then I'll just go back to the default setting back. Then you can save this thing as well. You can save. So let's probably want to share what you have with somebody. You can save and then send it either as a TXT file or CSV file or PubMed, or I think this and this. If you actually want to share it with somebody, say the best. Yeah. If you don't understand up to this point on how you can search using the basic I mean the simplest approach, and also using the Boolean operators, using the advanced functionality of the on-tree search engine, and using the mesh terms. Do you understand it? Sure, yeah. Are we together? Or are you confused?
We can confirm that by asking questions. Do we understand? Samuel or Lami, do you understand?
Yes, sir. We're grabbing it.
Please, in a sentence or two, can you explain or can you tell me what you understand by mesh terms? And how does it help you? Samuel.
Okay, I think that is a medical search. I can't remember the name of I say, but there we can use a unifying term to search instead of using some other I don't know how to put it, but a unifying term just to get the results for a
Yeah.
particular term. Yeah.
Okay. All right, you tried. Let me ask another question. Sadiq Omotoyosi, can you hear me?
I don't understand though.
Hey, and we're saying, do you understand? You cannot say I don't understand.
My network got cut off when you were explaining mesh, so I don't understand.
Okay. Continue. Family Oluwole.
So I presume what's the question?
The question is, what do you understand by mesh?
Okay, it is using a general term, a one term that you can link other similar terms to. Like there's a general term, then there are other words that are synonymous to it. So yeah.
Okay, thank you. Abib, do you want to continue? Or you still want us to probe deeper?
Yes. Well, I think probably we can continue.
All right.
So I may probably after this class for so deep, if you can probably watch this video and if you still don't understand, probably you can ask in the next class. So yeah. Yeah, I think we are done with the first task of how you can do the resource search. I mean, there are other ways that you can. I mean, there are lots of functionalities on NCBI. You know, I will always keep telling you this. So you should actually play around this with this database so that you can actually know it very well. I won't lie to you. In fact, I'm always uh, discovering new things on NCBI myself. In fact, there are some things, I mean, some link that I've never clicked before on NCBI. I don't even know how they work. So, I mean, but PubMed is actually very essential for you, especially if you want to be a researcher. You have to like read other people's journal, I mean, articles. You have to like read them, and yeah. And the one way you can actually probably use to search is to exploit all of these Boolean operators, I mean, using the right mesh terms and things like that. So you can also see that, I mean, when we did this search, let me show you. I mean, you know, the first thing that we did was to just copy all of these things verbatim. Then we came here, and we came here. Then So what we just did was to do this, right? Then we search. So if you actually come to this advanced, you actually see what is going on behind the hood. So this was where I just click. What row does pain? I have a sleep disorder. So you can see that in a way, the on-trace search engine it has splitted all of these things into, individual words, and it's looking for the mesh term, control vocabulary to actually represent. For row, I think it is still row and outfit. So it's actually breaking down things into the mesh terms so that it's actually doing the right thing. There are some times when probably this might work. So, and whenever you do something like this, probably this kind of search, you should always probably read the interpretation. How the on-trace search engine actually interpreted. This was what you entered, and this was the on-trace search engine interpreted. So it's encoded all of these things as this. And this was what was used to search. You will see that if I should actually copy probably this and put it and probably search for something here, I should still get the same thing. So 116 is still the same thing, right? It's still the same thing. I should go back 116. So there are some times when it's going to work, but always confirm whether it is actually interpreting it the right way, and then you can then modify it. Probably it's not using the right mesh term. Based on your opinion, you can probably edit this and then put the right mesh term so that you are getting the right information. I'm not sure whether you understand what it's talking about. So the second task we are going to be talking about is how you can search for sequences or weather protein sequences or the nucleotide sequences on NCBI. So again, we'll go to the NCBI homepage. So I'll just click on this logo. It's going to take us to the NCBI homepage. So the first thing we are going to look at is how to can probably let's start with nucleotide, like nucleotide. Can I see nucleotide? So I click on nucleotide, right? Let's probably want to. And always tell me one gene that we can probably search about. I think probably human cystic. I think I have an example here, so I'm just go with that. Fibrosis, transmembrane, conductive regulator. So I want to search for this. So this is the name of the gene, human cystic fibrosis transmembrane regulator. Let's just search for it. I don't know what to get for. Let's search. So no item found. So, and that's because it is expecting the name of the gene. So if we shorten this gene, it is CFTR. So that is the name of the gene. So, and if it is the human that you're interested in, I will just search for the general one, CFTR. So this one is cystic fibrosis transmembrane understand. So this is the name of the gene. So if we click on it, so we can see CFTR, CF transmembrane conductance. So we can see all the nucleotide sequences pertaining that particular gene. So you can see one is from the Orictolabus cuniculus CFTR. That's what you are looking for. Fine. And this one is, we have the one from Homo sapiens, Latus. I'm just going to probably limit this to Homo sapiens because we are just interested in the human. So C, FCL, let's say probably human. Let's see what will happen. So you can see we now have everything on Homo sapiens. So this is the first hit, that was written. And you can see from here, the first one is human cystic fibrosis gene exon 3. But this one is just, okay, let's even click on this gene CFCL. So to log this, let me click on this and then see. So when you click on this, let's see what will happen. So you have information about this that you can read up. So the gene name is CF transmembrane conduction regulator, and this is the symbol. So it has this gene ID. It is found in Homo sapien. It's telling you that this particular chain with this gene symbol was isolated from Homo sapiens. So the gene type is protein coding. Synonyms, so these are the other names. If you don't want to call it CF, very cool. Either refers to it as what CF, MRP7, ABC35, ABC, and then a lot of information. So the accession number, if you want to see the gene context, what are where exactly in the chromosome is it located? So it's saying if you go to chromosome number 38, so you should, and position number 14, you should be able to find it there. So, and then the accession number of that is this, and then annotation, information, then the chromosome that is found in chromosome number 7, rather. Sorry, chromosome number 7. So that's when you find this particular gene. So the genome assembly and things like that. But what we are interested in, so can just probably play around. We have a lot of it that was returned. So just click the one that you think is relevant to you. So let's probably, you are interested in the human cystic fibrosis gene, the exon 13. So I'm not sure whether you guys still remember your introns and exons. So, eukaryote genes are interrupted, by introns. So, and then all of those introns are going to be spliced out, and they will be there with exons, and the exons will be transported into your final protein. So that's probably you're interested in gene, the exon number 13. So this is the information about exon number 13 of the gene CFTR gene. And then, so you can see the locus name. So the locus is just the same as probably the name of the gene. So, and it's telling you that it is 47 base pair long. It is a DNA sequence. It is linear, and I think this was first reported, deposited November 1994. Then the definition, so we just trying to define the gene for you. The name, human cystic fibrosis gene exon 13. And this is the accession number. So I'm going to tell you about the accession number later. So the accession number is just like a unique identifier. So all the record that you see in probably your gene bank or like NCBI, they have a unique identifier that just see it as your matric number. No two students in UI can have the same matric number. So matric number is unique to individual. So I have my matric number in UI. Then nobody else is going to have that. So, so this is the fashion number. So it's saying that this is the fashion number 1. So it's possible that somebody probably an update is actually probably done on this particular gene, and then by the time you deposit that one, that update, that will be fashion number 2 as well. So every now and then, I told you that NCBI is well curated. So there are dedicated people that are always trying to check for correctness and for accuracy and things like that. So whenever they spot anything, and then they collect it, so they increase this fashion number. Or so far, they increase this fashion number. So, but I think the fashion number 1 of this is this one. It's possible that probably there is fashion number 2 already. I don't know. So, and the key word is cystic fibrosis mutations. So you can read on this. So this stuff that you are seeing, these 5 formats, we call it gene bank formats. And you can see it here. So gene bank formats. The gene bank formats. So this is how the gene bank format is represented. So from year to year, is information about, so we call it the metadata, section. So it's just telling you information about this particular gene. So from year to year. And then, so what I want to have in mind is the gene bank format is divided into three regions. The metadata format, and the metadata region is from years to year. Then you have the features region, which is from year to year. From years to year. And then you have the origin or the sequence. So, and the origin is from years to year. So those are the three sections. Within the gene bank format. The metadata format section, which is from years to year. The feature section, which is from years to year. And also the origin section or the sequence section, which is from years to year. So in the metadata section, which is from years to year, what is happening here is just describing the gene itself, whether if it is a protein sequence or a gene sequence or so it's just giving information about those sequences, where they are from, when they were first described or deposited, the organism that you isolated it from, whether the articles that the gene was first reported, things like that, and the authors, the person that actually discovered or deposited it. So you find the information again. You can see everything here. So you find it here in the metadata section. And then again, so in the feature section, so that one is just telling you, like, just see it as the annotation. So this is, so by the time we actually get to file format, you actually see that most of this by the time we start our, how should I even put it? Annotation, gene annotation. So we'll still replaces the gene bank format again. So, but what this one is just telling you is that, we said we have this thing is 47 base pair long and it's DNA sequences. So it's telling you the source. So from here, so it's just telling you the source. That's okay. Like 1 to 47, the organism is Homo sapien. The molecular type is genomic DNA and not, RNA. So the database references is this, and the map is 7Q. Like, I don't know what this one is talking about. But this, so this one is telling you, okay, if it goes to this position, position 1 to 7, so it's a gene, so you will find it is CFTR. And the exon also is what? CFTR. So this is a sequence consists of a new frame shift mutation in which the DNA is deleted. So sometimes, so the reason why you are seeing this, and then the coding sequence is telling you about information about the gene, the exon, and also the coding sequence. You can see that they are, all of these things are the same thing, and that is because it's an exon. And the exon also is what will be translated, you can see it as a gene, and then it's also a coding sequence. All exons are coding sequence. So that is what is happening here. So it's just trying to tell you that if you go to this particular region, this particular position in my sequence, so you will find a gene named CFTR. And because, is this an exon also? It's trying to say, okay, is this an exon? Is it coding sequence? Yeah, it's also a coding sequence. It's a coding sequence, and this is the information about it. And this is a note. It's just about, it's describing what is happening in this particular position. And is it a coding sequence? Yes, it's a coding sequence. And it's also telling you that from position 1 to 44, it's the coding region, coding sequence region. And the gene is this. Codon starts at, probably position number 3. So that's when so that's why you are saying this one to be 4, to be 1 to 44. So because it started, it's just the length of the position is 44 base pair long, and that's because the reading frame starts at position number 3, and not position number 1, and position number 2, position number 3. So if you add this 3 to this 44, you should get 47. So the coding sequence by itself starts from this, from position number 3 up to position number 7, but the length is 44. And the gene and the exons they both have the same length, 144. Most of the time, your gene and exon, so they are going to be the same if you are talking about exon, But let's probably are talking about the entire gene itself. Let's say probably we are just talking about the CFTR without the exon. So the gene and the exon, they will be different. So they will be different because the gene is just the entire one and the exons are just the specific region. You know, an exon can be a subset of a gene. So in eukaryotes. So, but if we are talking about prokaryotes, the gene and the exon information, so they will always be the same thing. They will always be the same thing. So the reason why, because this is an eukaryote, but this is why they are the same information, I mean, they are the same length, is because this gene is the same as an exon. And but the coding sequence, I mean, when it starts, I mean, when the ribosome is going to start, I mean, translation is going to be from the position of it. And you can see, so this is the sequence itself. You can see. So if you do this thing, so the length should be fixed on base pair. So let's now probably wants to change it. So like I said, this is represented in what? In inbound format, but you can change the format to faster format by just clicking this. So if you click on faster, you can see. So you now have it in faster format. And then you can download if you want and download this. And then you click on this and then create file. You can see that I now have my information. You can see I've downloaded it. So you can change it back to GenBank formats if you want. And then if you want to download that one also, you can download it. File, GenBank formats, you can do that. And if you do this, you can see the same information that I have it here as well. Do you understand up to this point? Or am I boring you with a lot of information? Is this an information followed on?
I think you are fine. I'm not sure there should be information about it.
So deep. No.
Yeah.
Please, do you have any question at this point?
Yes,
Do you have any question at this point? I can't hear you.
Yeah, don't hear her too.
Bola Tito, is everything fine? Bridget, are we together?
Alima Sulaiman, did you
Yes,
get it?
sir.
Bola Tito, Bridget.
Thank you.
Yes,
Bola
I got
Tito,
it.
did you
It's
get
fine.
it?
Joy, Joy
Joy,
and
yes.
Joyce.
Yeah, Joy.
Joy and Joyce, are we together?
Yes, I got it.
Samuel,
I don't have any question.
sorry, can you
You come on to your phone.
type?
I can't hear you. I guess your phone is ringing or something. No, your network is bad.
Samuel, are we together?
Yeah,
Samuel.
yeah. Thank you, sir. You are good.
Shegun. Okay. T. I don't know who T is. Who T be? Mr. T, are we together? So don't worry. I mean, we'll still talk about the gene bank format. I think that should be the next class when we start talking
Yeah, I
about.
think you went a little ahead with the file format.
Yeah. Okay. Yes. All right. I think we can go ahead. Yeah. So if you want to run Blast, we'll still teach you Blast. In fact, there are a lot of things that you can do. You can run Blast directly from here if you want. You know, regarding the sequence. We'll still teach you Blast. So Blast, so there are so many ways that you can use to find the sequence. So that if you want to find a sequence, you can use a sequence to find a sequence. So you can also use keyword. I mean, but the one that we are seeing here, so if I should go back to my slide, so probably I should be able to describe that better. Okay. So we can say I said using the keyword, the gene or protein name. So you can use this one, I mean, the gene or protein names to search. So that was what I just did. You can use the accession number. In fact, this is the most specific. If you know the accession number of a particular gene, is the most specific. So, like I said, let's probably, I was telling you about every student in UI having a matric number that is unique to individual students, right? So no two person can actually have the same matric number. So let's probably, I'm just interested. I just want to like probably call on Uyilami Samuel. It's possible that we have a lot of people that, have the name, that answers the name Uyilami Samuel. It's possible. So if you just probably call somebody by Uyilami Samuel, a lot of people might actually show up that are bearing the same Uyilami Samuel. But let's probably are just interested in a particular Uyilami Samuel. So just refer to that person through his or her, matric number. And then that is going to be specific. So let's probably I do something. Let's probably I did something wrong, and then I want to be expelled. My name is Abibadeni, and I say, okay, Abibadeni has been expelled from school. So there could be a lot of Abibadeni, but we can put Abibadeni with the matric number dash dash dash. So I will know that okay, I think they are talking about me. So it is my own Abibadeni that is being expelled from school. So accession number is still the best way to search for anything. So let me just show. Let's look for let's so this is an accession number. You can just probably copy this. Let's say I actually want to get an information regarding this particular one. So I will just do this. Remember, so if it is gene that you are interested in, if it is protein rather that you're interested in, you can just probably click on this. But let's even do it on gene, or you can do it on the protein. I'll just do it on gene, and then use this new and search. So you can see, they didn't give me all of those lists. They just took me directly to this particular sequence, which is this. A lot of information. Yeah, a lot of information. So the gene contests and everything. You can then start reading the expression. So you can see a lot of information here. In fact, you can even publish a lot just by doing this. Because so this one is telling you that okay, this gene, it is expressed. Just because you have a gene does not mean you are expressing it. So having a gene does not mean expression. So in adrenaline, one thing that I want you to know is that eukaryotic cells, we are multicellular cells, right? And we have the same, despite the fact that we are multicellular cells, our cells have the same DNA information, like the same DNA information, but we have differential gene expression. So what is actually making your epithelial cells look like epithelial cells and not to look like probably skeletal, is it musculoskeletal cell? I don't know what you guys call it. It's because of differential gene expression. So some genes are expressed while the other ones are depressed. In the muscle cells, some genes are expressed and some genes are repressed. So this one, so all of this adrenal stuff, 20 weeks, 20 minutes, so all of this that you're saying. So it's saying that okay, this particular gene, so it's not expressing the adrenal gland, I don't know what adrenal cell in 10 weeks when you are 10-week old. So it's also expressed, But the first expression is going to start probably at 10 weeks. So you can see, it's just telling you, okay, this particular gene that we're talking about. So you can see that it is highly expressed in the intestine when you are probably 20-week old. So, or 20-week, I don't know whether this has to do with fetus, okay, fetal development. So this gene, I think it is very critical in fetal development. And you can see when expression starts. So expression starts in the heart, when it is 10 weeks. when you are just probably 10 weeks to I mean, fetal development, as you can see. So the expression is very low, you can see. And the expression is highest in what in the intestine. So in the intestine, the expression is going to start around what 10 weeks, and it's going to peak when it is what 20 weeks. So that's what is just going on here. So a lot of information, and by the time you just probably read on this gene contents, you can then start probably even writing papers and say, okay, we want to even describe, I mean, this particular gene in full. You can probably, I mean, these are the articles that you can cite, or you can even read more about the pharmacokinetic environment of luminance in children with cystic fibrosis. So this particular, I mean, gene, I think is implicated in cystic fibrosis. So, yeah, so if there is any mutation in it, probably the person is going to have, I mean, the cystic fibrosis disorder. You can see, so there are a lot of information that you can just read up regarding this. So I got to this page by clicking on gene, but if I click on nucleotide, let's see how everything will look like. So we can see, and now the gene long format that I want. So we can see it here, right? It's even saying source test human Italian patients. So this particular, so gene was isolated from a woman of Italian descent or Italian patient. I don't know if it's Italian descent. It's an Italian patient, right? So you can see. So I think probably this is the first report of cystic fibrosis gene. I don't know. So, and this is 1994. So, like I said, accession number is still the best way to what? It's the best way to get a particular gene sequence or nucleotide sequence or protein sequence. And it's when you use the accession number. But let's probably you don't have the accession number. So all you just have to do is probably just type this broad generic name. Just type it here. And then just start reading and then pick the one that applies to you. So just click the one that applies to you. And then, in fact, you can just even download this. Download this. You can download the accession number list or the GI list. So this thing, let me see what will be reported first before I say anything. Because I think I've used this functionality before. So that was a time I was trying to like get all sequences pertaining a particular gene. And because I wanted to do like a multiple sequence alignment. You can see this is a lot of fun. So what is going to happen is it's going to like download all of the accession number here. So you can now probably take this and put it on your command line, then download the sequences pertaining that. I mean, the sequence corresponds to each of those accession number. So this is a lot of file, but let's see what will be reported first. This is a lot of information. So let's continue. Okay, I think this is try. Okay. This one is okay, accession list. I think session. So you can download this accession list. I mean, let's probably want to get all of those information. So instead of doing this, I think I did it wrong. So let's probably you want to download all of the sequences. I mean, on your command line. So you can just copy all of this. I mean, come to your command line. So you can come to your command line. Then you can create a test. I mean, nano TST. I mean, nano session. and then paste this. Then you can now write a loop to fetch all of the sequences. So there's a way you can write a loop and then fetch all of these corresponding sequences that belongs to this thing. So using fetch. So the reason I won't show you this is because we are just to teach you how you can download or fetch sequences from NCBI from the command line. So when we get to that, I think in the next two topics, so we actually probably will visit this functionality again. So we'll just say, okay, we'll go to NCBI, search for CFTR gene, then get all of the accession number corresponding to this particular gene, and then fetch the corresponding genomes and these corresponding sequences of those accession number, and then do probably multiple sequence alignment or consult a genetic tree based on the sequences. So I wanted to probably take note of this. It's a functionality you will actually probably come across, come to use in subsequent in future class. So please, I wanted to probably take notes of this particular one. So that by the time you say, okay, do this thing, you understand how I'm not going to do it. So what I just did was it was on summary before. So what I just did was so this was how everything looked like before. Then I just clicked on a session list. So when you click on the session list, you have this information. So just copy all of this, transfer it to your terminal, and then write a code that will then fetch everything. So the code is just going to it's going to be a for loop, and then you fetch all of the genome, and then you can now start working on the genome. So that is that about that. So this one is CFTR, but let's say we are interested in Homo sapiens. Like I said, Homo sapiens. Human, I'll just put human. Homo sapiens. Yeah. So this is where we are. So just pick the one that you want. So this is if you are not using the accession number. So you have to then start looking which accession number which particular one are you interested in. But if you know the accession number is still the best way to actually get like a particular sequence. So you can also search against the protein database. So if you do that, it's still going to probably give you the same information, but so this one is just going to be a translated version of that. So and then let's say click on which one should we click on? Yeah, we should just click on this. Let me just click on this. Just for explanation. And you can see still a gene bank format, but you can see the same information. And you can see this is the metadata section, and this is the feature section. So the feature section, and we can see the faster the sequence. So we want to change this to a faster format. You can do this by clicking on this. Then you have the faster file. Then you can download if you want. Just click on this. And if it is another format that you want, any format that you want, just download it and click on this. Then it's going to be downloaded. You can see that's downloaded. And let's say we want to run a blast directly from here. You can just click on this run blast. And then it's just going to look, find related sequences to this particular one. And by the time you run blast, I'm just going to go with the default when we get to blast proper. Yeah. So blast is working. So the page will be automatically updated in 2 seconds. So this actually, just play around. So when you get home and when you watch this video, play around. I've not taught you everything, like I said, but I think with this one, you should be able to do majority of these things that will be asking to do in subsequent classes. Yeah, please, do we have any question? As you can see, we now have our search, our blast results. You can see we have like a 8 from human, domestic to sheep, this thing. So that means this our particular gene is not just only found in human, it's also found in other organism. So and it has found like an autologous gene. Yeah. Do you have any question of it? Any question of it? Yeah. So if you don't have any question, I think I'm done for today. So, please, do you have any question before we call it a day? Please, do you understand? Hello.